Great. Well, thank you very much, and thank you, Jack, for that wonderful talk, setting the whole scene. I'm going to try and give a sense from the, if you like, from the inside looking out. What can we learn about the history of life, the history of our planet, how things have changed over this 4 billion years from what we know about the patterns of life today? It's a strange question, but why is life the way it is? This is a tree of life that you'll still find in most of the textbooks. It's known as the 3 domains tree of life. We are across there with Carl Woese, who was the person who really put this together, and this goes back to about 1990. We are eukaryotes, which is to say we have a nucleus where we put our DNA. And you can see, I hope, that animals, plants, fungi, and so on are clustered quite close together there. Across at the other side, we have the bacteria, and then this other group in the middle known as the archaea. Now, this is the lengths of these branches gives an indication of the amount of genetic variation in these groups. And there's one thing which seems to me, I've got a rather challenging title there, information alone can't explain the tree of life. The bacteria and the archaea, these 2 large groups with a tremendous amount of genetic variation in them, in terms of their biochemistry, in terms of their genetics, they've explored evolutionary space in a way that we as eukaryotes seem stunted in comparison. But in their morphology, they're rather small and don't look like much down even an electron microscope. We know there's a tremendous amount of structure there at the molecular level, but as in terms of what you can see, there's not much there. So there's something about that tree of life down that green branch leading to eukaryotes that gave rise to the complexity that we see around us. But something else in these 2 other groups, the bacteria and the archaea, that seems to have been holding them back. It wasn't that they didn't explore genetic space, they did that a lot. There's something that was constraining them, perhaps something structural. This is my take on the tree of life from the point of view of a bacterium. As Jack was saying, that we can see signs of bacteria in the fossil record going back practically 4 billion years, 3.5 to 4 billion years. The reason I say we see signs of them is they look an awful lot like modern bacteria that we'd recognize today. And so in some sense, they simply haven't changed in their morphology. And we can tell from a lot of the geochemistry in the rocks that the kind of biochemistry that they do also has been rather similar over billions of years. And at just one point in the history of life, these large, complex eukaryotic cells seem to have emerged. Only once. Perhaps it happened on many occasions, it's just we only have evidence for it happening just once. So what I'd like to talk about then is, well, what are the forces that constrain the evolution of bacteria? Why don't they become kind of larger and more complicated? Why don't we see creatures of the level of complexity of even a flea composed, say, of bacterial cells rather than our own type of eukaryotic cells? How did complex cells escape from these constraints? And would it be at all similar on other planets, or is life on Earth just a particular case, constrained by forces that might not apply anywhere else? So we have to immediately frame the question, well, what is life? And this was framed most compellingly, perhaps, by Erwin Schrödinger in a famous book, which was 75 years ago this year, called What is Life? And really there were 2 themes in that book. One was about genes and information. And he was the first person to talk about a code script. He was really the first person to see biology in terms of information and encryption, in terms of codes. This was during the Second World War, 1944. It was when this kind of encryption was in the air. And he talked in quite modern terms about genetics. This was before the structure of DNA was known. That was 1953. He also talked about energy, or really what he called entropy, negative entropy. And here his language is perhaps a little more archaic to us today. He talked about life continually sucking orderliness from its environment. And it's a little difficult to know quite what he had in mind by that. But he did have a footnote where he said, if I'd been catering for physicists alone, I should have let the discussion turn on free energy instead. Well, what is free energy? The way we get our energy is extraordinarily complex. These are the mitochondria inside our own cells, and the membranes that you can see, the darker blue membranes in the middle, is where cell respiration is taking place. So we are burning food in oxygen across those membranes. But it doesn't have to be food. It does for us, but if you think about bacteria, they could be taking hydrogen or hydrogen sulfide or anything like that from the environment, and they don't have to burn it in oxygen. It could be in nitrate or sulfate or something like that. This is roughly what's going on. So the blue band across the bottom there is supposed to represent one of those membranes. The red balls, these are extremely sophisticated protein complexes inside those membranes. Effectively what we have is a flow of electrons, in our case coming from food to oxygen, in the membrane. So we have a current of electrons within the membrane flowing to oxygen. That current of electrons is powering the pumping of protons across the membrane. So protons are the nuclei, positively charged nuclei of hydrogen atoms, and they are flowing back through what we might call a turbine in the membrane, what's known as the ATP synthase. ATP is usually called the universal energy currency. All life tends to use that. In fact, all life tends to use something very similar to this system, which you could think of as very analogous to a hydroelectric power system, where the proton reservoir on one side of the membrane is equivalent to the reservoir, the membrane is equivalent to the dam itself, and then the electrical turbine, well, this is the ATP synthase. The structure of this was determined largely by Sir John Walker, who got the Nobel Prize for that. It's a nanoscopic rotating motor. It's an extraordinary machine. And I'm just really showing that to say this is universally conserved across life, and yet it's such a sophisticated machine. It is plainly a product of genes and natural selection and evolution over a period of time. This says nothing about the origin of life. But it does point us to say, well, the processes of having proton gradients across membranes, it's so universal. It's as universal as the genetic code itself. What is it telling us about life? Could some of these structural constraints on bacteria reflect something to do with the way that energy works in cells? The person who discovered that more than anyone else was Peter Mitchell and his lifelong collaborator Jennifer Moyle. It was really Jennifer Moyle who did all the experiments. It was Mitchell who had this particular idea. And it really was a counterintuitive idea. Not since Darwin has biology come up with an idea as counterintuitive as those of the great physicists, Einstein, Heisenberg, or Schrödinger. That was a lovely quote from Leslie Orgel, one of the great scientists working on the origin of life. Now, Mitchell himself was at a conference in Moscow in 1957, before he even put this theory forward, 4 years before that. And there's a lovely quote from him at that conference, which resonates with me a lot today. He said, "I cannot consider the organism," and when I say organism here, think about bacteria, don't think about ourselves, "without its environment. From a formal point of view, the two may be regarded as equivalent phases between which dynamic contact is maintained by the membranes that separate and link them." So he's talking about a membrane which is just a few millionths of a millimeter thick. It's very, very thin. On one side is the cell, the organism. On the other side is the outside world, the environment. And here we have this membrane, and he's describing them as equivalent phases on either side of that membrane. It's a radically different way of seeing life. It's trying to define life as part of the environment rather than as something separate to the environment. This was what he was thinking about. He wondered, well, how do bacteria keep themselves different to the external environment? Why are these two phases different? And it's obvious that they have to recognize particular molecules and pump them out or bring them in. And if you're going to do that, it's going to cost some energy. If you're physically pumping things outside the cell, and in this case I'm showing protons, then if you were to allow that to reverse and allow them to come in again, then you might, in principle, be able to conserve some of that energy and put it to use. So this is the free energy that Schrödinger was talking about, the way that it actually works in cells, as first proposed by Peter Mitchell in 1961, and which turned out to be the truth. He received the Nobel Prize in 1978 for that work. So this is how even the simplest cells seem to operate. So methanogens, they produce methane as a waste product. This is one of the issues with global warming. Actually, cows are producing methane from the other end, but I just rather liked that picture. The reason they're producing methane is that they have methanogens in their intestines. And methanogens live from this very simple reaction between carbon dioxide and hydrogen gas. Two of the components of the early atmosphere that Jack Szostak mentioned that were probably long-term components of that early atmosphere. They live from that reaction alone between carbon dioxide and hydrogen to produce methane as a waste product. And they get some energy from that reaction. And that energy is enough to drive all of their biochemistry. But they don't do it in a very straightforward way. I'm not going to go into the details of this, but essentially the HDR, that red circle there, this is an enzyme that takes the 2 electrons from hydrogen. And one of them, it pushes one way to do a really difficult reaction that it doesn't want to do, but it's coupled it to a reaction that really wants to happen spontaneously. So the two have to happen together, and that difficult reaction is coupled to a very easy reaction. An exergonic is coupled to an endogonic reaction. That reduces a protein called ferredoxin, an iron-sulfur protein. Again, this is universally found across all of life. That passes its electrons onto carbon dioxide to fix carbon dioxide, to convert it into organic molecules like the methyl group that you see there, the CH3 group. The methyl group is transferred from one enzyme to another enzyme, and then it picks up those electrons and it's lost as methane. So the only thing you need to take home from that is nothing is concerned. The electrons are taken from hydrogen, they're put onto CO2, and the waste product is methane. It can go round and round like this forever and forever, and the only thing that's conserved is a proton gradient across a membrane. So that begs the question, well, what do they do with that proton gradient across the membrane? And the answer is, well, they use it for, there's a protein there sitting in the membrane called the energy-converting hydrogenase. And what that does is it uses this flow of protons coming back through the membrane in the same way that Peter Mitchell was talking about to drive the fixation of CO2. It's converting CO2 into organic molecules and the beginnings of metabolism. Here we have a proton gradient doing all the work. The question is, are there environments on the early Earth or the Earth today where we have natural proton gradients, geologically sustained proton gradients that might potentially do the same job? Well, there are. This is perhaps the most famous environment. There are probably other environments that might do this as well. These are known as alkaline hydrothermal vents. They were discovered in the year 2000 by Deb Kelly at the University of Washington. And this is Lost City, the vent field that she discovered. It doesn't look much like the familiar types of hydrothermal vents that most people are familiar with, the black smokers where you see lots of smoke coming out of the top. These almost seem deserted, but they are active vents. There are fluids moving through them, and there are bacteria and archaea that live in those environments. This environment had been predicted and worked on for many years by a guy called Mike Russell. And here was a Nature article from 2006, I think, presenting him as what they called Naissance man. They dressed him up as Renaissance man, as Erasmus or something, and Naissance as in the birth of life. Now, Mike Russell's ideas are somewhat controversial. I don't think necessarily that Jack Szostak would agree with them, but the essence of what he said is that in these environments you have high concentrations of hydrogen. The early oceans almost certainly had rather more carbon dioxide in them than today. And you can see on the right-hand side there that this is a series of interconnected pores. And probably then, again, we don't know for sure, the chemistry of the oceans was different, and there were probably more reactive minerals, iron-sulfur minerals, in those walls. And so what you would have would be alkaline fluids with hydrogen gas percolating through a reactive labyrinth of micropores. That's the idea. It's an electrochemical flow reactor, in effect. This is the conception, is that you have a pore. On the inside, it's alkaline with the alkaline hydrothermal fluids. On the outside, there's a relatively high proton concentration. And on the right-hand side there, you can see Mitchell's conception of a cell. So the topology is very similar. You have protons outside, hydroxide ions inside, and a barrier between the two. But that is simply an analogy. It's a big question. Can we somehow go from one to the other? Is there any way? that we could think of, that in the absence of genes, in the absence of proteins, in the absence of really organic molecules at all, that a proton gradient across a barrier might be useful for something? Well, we don't know. We can put ideas forward and we can try and test those ideas, and that's what I've been trying to do for a while. This is the slide you've already seen, showing what cells use these proton gradients for. On the right-hand side there is a basic structure of this protein, the energy-converting hydrogenase. It's an iron-sulfur protein, which means that it's got these little clusters of iron and sulfur atoms which are doing the chemistry. And those sulfurs, those clusters, you find them in many different types of hydrothermal environment. What you can see is the reaction between hydrogen, it's reducing ferredoxin. It should not happen. That reaction is the energetics of it are not favorable. It really shouldn't happen. It's the pH gradient which seems to be driving it. Those iron-sulfur clusters, their reactivity depends on whether there's protons there or not. And so the continuous flow through of protons is changing the immediate chemical environment at that interface. And it makes a reaction possible that under normal conditions, just in a test tube, doesn't happen. Why? Well, it's in part because the reactivity of both hydrogen and carbon dioxide and these iron-sulfur clusters depend on the proton concentration. I won't go into the details here, but you can see at pH 7 or any other pH you choose, it's uphill. That's really all you need to take home from that. Hydrogen doesn't want to push its electrons onto CO2 to make organic molecules, in that case, formaldehyde. It's steeply uphill and it simply doesn't happen. That's why it's proved difficult to strip CO2 out of the atmosphere and convert it into synthetic gasoline. It's doable, but it's not very easy. But the reactivity of hydrogen is much more reactive in alkaline solutions. It really now wants to push its electrons onto something else. And carbon dioxide, it's much more reactive in mildly acidic conditions. It now wants to collect those electrons from somewhere else. And so if you had hydrogen at pH 11, as we do in these vents, and CO2 at pH 6, as we would have had in the early oceans, then in principle, it should actually work. But you can't have mixing of solutions. What you would need would be some kind of barrier, and that's what we see in these type of vents. So in principle, that pattern there where you have CO2 in acid waters percolating through, hydrogen in alkaline waters percolating through, and a barrier between the two, which is either transmitting protons or electrons, in principle, that could work. And it's a testable proposition, and we've been trying to build microfluidic reactors and larger reactors to do that, to see if we can have steep proton gradients across. So these are showing inside a microfluidic reactor, the greeny yellow color is the acid, the blue is the alkali. And you can see that there's a very sharp distinction between the two, and the barrier itself is relatively acid, so the protons are crossing that barrier relatively easily. We've had, I would say, limited success. This is not something that I'm very happy about. Certainly, it's much, much easier to do this chemistry if you start with cyanide. But from my point of view, ironically, as Jack said, we don't associate cyanide with life. We associate CO2 and hydrogen with life. And so, does it work? This is the difficulty, is to demonstrate in the lab that we can actually do those reactions. We've had some success, but we have difficulties in repeating it on a regular basis. And so I won't talk through that, but these are very, very simple organic molecules: formaldehyde, formate, even carbon monoxide. But we've also had some interesting other results, which is to say, we found that if we start with these very simple molecules, and in this case I'm showing cysteine, which is an amino acid, or methionine, but we can also do it even with things like bicarbonate ions, they interact with iron and sulfide in these environments, and they form spontaneously the same iron-sulfur clusters that we find in modern proteins today. This is at the right-hand side, Mossbauer spectroscopy showing 4 Fe, 4S clusters and 2 Fe, 2S clusters. These are the type of cluster you can see there that we still find in modern proteins today. So simple organic molecules will form exactly the same clusters that we see in cells today. And they have exactly this pH dependent in their reactivity that I've been talking about. So they get down to minus 600 millivolts at about pH 11. They really should be pushing electrons onto CO2, and we're still working on that. So I want to broaden this out again. I don't want to say any more about the origin of life, but this kind of environment where Lost City was formed is this process called serpentinization that Jack mentioned. It's a reaction effectively between rock and water, between the kind of minerals that we tend to find in the upper mantle of the Earth, like olivine, for example. If you have a lump of olivine and were to put it in a bucket of water, it will react with that water. You get one bubble of hydrogen in about a week's time. It's not an experiment that I really encourage. But down at the higher temperatures and pressures in the oceanic crust, so beneath the ocean, this reaction is happening today to produce alkaline hydrothermal fluids bubbling in hydrogen gas that comes back up, percolates back up through the crust, and then forms these vents at the surface. We know that similar processes are happening in our own solar system, so almost certainly on Enceladus. The plumes on Enceladus are strongly alkaline. They're about pH 9. In fact, I've said 11 there. I think they're not quite as alkaline as 11. There are organic molecules there, quite a lot of organic molecules and hydrogen gas. So we see this same association. We don't know of any other conditions really other than serpentinization that would produce such alkaline fluids and hydrogen gas in such large amounts. So we can be fairly sure that that's producing the hydrogen on Enceladus. We're fairly sure on Europa we see similar processes going on, and even the traces of methane that have been detected occasionally and sometimes not on Mars also probably are a result of serpentinization on Mars. So this is a process that is undoubtedly widespread. How much it contributes to the origin of life, we don't know, but it certainly is providing reactants like hydrogen at the very least. How common would it be? How many wet, rocky, Earth-like planets are there? Again, we don't know for sure, but this is very exciting times. And it seems as if there could be in the order of tens of billions of exoplanets in the Milky Way alone that have sufficiently Earth-like conditions defined in those terms. Olivine is ubiquitous in interstellar dust. So is water. And CO2 is common in planetary atmospheres. So these conditions would seem to be common across the universe as we know it. So just for the last 5 minutes or so, I'd like to talk about the second half of this question. Why did life get stuck in a rut? Now, this depends in part on what I've said already, which is to say, all bacteria and all archaea and all life as we know it is powered by proton gradients across membranes. It seems to facilitate a reaction between hydrogen and carbon dioxide, which is the reaction that basically all cells use to live, even photosynthetic cells, where they're getting their energy from the sun. They're using that energy to split water and take the hydrogen out of water, and they're then reacting hydrogen with CO2 in exactly the same way as the methanogens do. It's the same reaction at the heart, even of photosynthesis. So, can that reaction constrain life? Can it explain why bacteria got stuck in a rut and remained relatively small, simple cells? It's what one of the great evolutionary biologists of the 20th century, John Maynard Smith, described as an evolutionary scandal. This idea that all complex life is composed of one particular cell type. If you look at plant cells or animal cells down a microscope, apart from the chloroplasts that do the photosynthesis, otherwise they're almost exactly the same. We share the nucleus, we share a whole series of different structures, cell structures like the endoplasmic reticulum, things like the mitochondria where energy production is going on, a dynamic cytoskeleton, motor proteins that move around the inside of the cell. We share all of this with plants or with fungi or with amoeba. So we know from that we share a common ancestor with plants and with algae and so on. And that common ancestor was already a complex cell. We can be fairly certain about that looked a lot like a modern eukaryote. And we know as well that bacteria don't seem to evolve any of those traits. They make a start very often, but they don't really do the same things that eukaryotes do. So the scandal, in John Maynard Smith's terms, is that if all of those traits evolve step by natural selection, and each small step offers an advantage, then why did none of them evolve in bacteria? Well, there's no question that they evolved step by natural selection and that each step offered an advantage. There's no doubting that. But it then becomes an interesting question, what stopped bacteria from having the same advantages that eukaryotes might have done? We can see part of the answer from this tree of life. This is just eukaryotes in this case. So these are the cells I'm talking about with a nucleus. And I like this particular slide from Eugene Koonin from a few years ago now because it's got a black hole, it looks like a black hole to me, right at the center of it. And you can see there are 5 what are known as supergroups of eukaryotes. These are not the traditional kingdoms of plants and animals and fungi and so on. These are names that are probably not familiar to most people: chromalvulates, rhizaria, uniconts, excavates, and so on. Most of the variation across, excuse me, across the eukaryotic tree comes at the level of single cells that you would see in those different groups. We are uniconts, by the way. I rather like that term. I certainly feel like a unicont, especially early in the morning. So the nice thing about that is the center of that tree is this black hole. That is the common ancestor of all eukaryotes. And it shared all of these traits that I've been talking about. But what do we compare it with? With bacteria. And bacteria have none of those traits. And so there really is a kind of black hole at the heart of biology. The common ancestor of eukaryotes was not a million miles away from this cell. This is actually euglena. It has beautiful chloroplasts, but apart from those chloroplasts, it's almost representative of what that common ancestor might have looked like. This is really pond scum in most people's eyes, but at the level of electron microscopy, it's a beautiful cell. And on the right-hand side is a relatively complex bacterium called planktomycetes, which has a small structure which is being compared with a nucleus. It's a compartment where the DNA is, but it doesn't seem to behave like a nucleus in many other ways. The reason I show it is this is where this gulf is in complexity between one of the more complex prokaryotes, a bacterium, and really a very bog-standard alga that you would find in any pond. There's a tremendous distance there, and we don't know how any of it evolved. So here's a problem for the young generation in the audience here. We have lots of ideas as to how these things evolved, and they're testable ideas sometimes, but we can't agree among ourselves, and we don't know who's right, and it's a difficult thing to try and prove, and this is something that I think the young generation will solve these problems. The way I think about it is particularly to do with this group of what are known as archezoa. So these are mostly parasites, but these are relatively simple cells that do not have mitochondria. And for a long time, it was thought that perhaps they never did. Perhaps these were living fossils, evolutionary intermediates on the trail from bacterial level of complexity to more complex eukaryotes. Well, it turns out that they're not. It turns out that they all do have mitochondria, just not as we really know them, or else they lost them fairly recently in the case of Monocerca monoides there. They've specialized them for other purposes, and they're now known as hydrogenosomes or mitosomes. So they're doing other jobs than energy production for these cells. But what that tells us is that this common ancestor of all eukaryotes already had mitochondria. And so we wonder, well, what kind of cell acquired the mitochondria? Well, this again, there's been tremendous work going on with environmental metagenomics, which is to say you go around and you take samples from the muds in all kinds of different environments. This particular environment was Loki's Castle, which is a hydrothermal system between Norway and Greenland, and sequence everything that's in the mud. So we haven't seen what's in there. Nobody, to my knowledge, has yet cultured anything, or these particular cells. But you can see in the pastel shades over there, what are known as the Loki archaeota. They were discovered at Loki's Castle. And the eukaryotes are that little red triangle there. And around them, these are other groups, what are now known as the Asgard superphylum, as the Norse home of the gods. So we see the Heimdall archaeota and the Odin archaeota and the Thor archaeota. They're all very closely related to eukaryotes. So it seems as if the cell that acquired the mitochondria was probably an archaeon that was something like that, but we don't know what it looked like. It's rather tantalizing. It may have looked something a little bit like this. These are the only known example of bacteria with bacteria living inside them. These are actually cyanobacteria, and they've got a couple of bacteria living within them. Nobody really knows what they are. This was discovered back in 1979, and to my knowledge, nobody has ever been back to this particular stream to work out what on earth these cells are actually doing there. I've been meaning to do it for a while, and it's one of those things that's very frustrating. I've still never made it. Why would that make a difference? Well, those bacteria living inside are cells, they're populations of cells. They can compete among themselves to get into the next generation of cells. And when they do that, they will tend to throw away things they don't need to be more competitive themselves. Genes that are no use to them here, they just get rid of them. And so over a long period of time, we know this from many examples of bacteria living in our own type of cells, eukaryotic cells. You can see the range of genome sizes at the top of free living bacteria. In fact, now it goes up to about 12 megabases, so slightly beyond the end of that graph. But down, obligate symbionts, these are the parasites and the cells that are living inside other cells, the endosymbionts. They have genome sizes down at 1 megabase of DNA, or even less than that. And these are some examples: Buchnera, Wolbachia, Carcinella. These are bacterial parasites of eukaryotic cells. So we know that they throw away genes that they don't need and don't like. And the extreme end point of this was the mitochondria. This is the image you already saw of the mitochondria. You might know that mitochondria always have a genome of their own, the mitochondrial genome. It's been really important in trying to, as we'll hear in the next lecture, in trying to decipher human evolution. What happened? When did different human species branch off from each other? Did they branch off from each other? What was going on? I hope that Donald will tell us something about that. But from a deeper evolutionary point of view, they always have a genome, and that genome controls respiration. So they've whittled it down to only the genes that you need to control respiration. And the few examples of giant bacteria that we see, so there's one here, this is bigger than most eukaryotic cells, it has tens of thousands of copies of its complete genome. It seems as if you want to scale up, you just need more genes to do the organization, you might say. And in the case of giant bacteria, they have to just copy their whole genome, and so everyone is exactly the same, and they're full 3 megabases of DNA, that's 3,000 kilobases of DNA. And on the right is, in the blue is the nucleus, and all the green dots, those are the mitochondrial genomes. They're tiny genomes, but they're needed to control respiration. And so there's been a change in the structure from trying to scale up something where everything's identical, so it's symmetric, those genomes are symmetric, to an asymmetry, where a massive nuclear genome with all the raw material for natural selection to act on is supported energetically by these tiny mitochondrial genomes. This is the long-term outcome of having an endosymbiosis between bacteria. It broke the structure of bacterial cells and replaced it with an asymmetry, which allows, this was some work done with Bill Martin some years ago now, but it just shows at the bottom there in red, these are the energy per genome or per gene availability in 3 different bacterial species, including those giant ones. And then in a couple of eukaryotes, Euglena, which I showed you the picture of, and an amoeba proteus, that's a log scale. That's 3 or 4 orders of magnitude difference in the energy available for genes. That doesn't mean to say you have 10,000 or 100,000 more genes. It means that from each gene, you could have 10,000 times as much protein synthesis. That's the real energy cost. You could have more genes and more protein synthesis from them. It could be anywhere in between. And so that black hole that I talked about, we're starting with bacterial level of complexity. We see an endosymbiosis between bacteria and other bacteria. We see a common ancestor of eukaryotes, which is a large complex cell. There are no known surviving evolutionary intermediates between them. Everything died out for whatever reason it might have been. That's really the work of the next generation. We only have ideas. What we need here is hard science. But it can begin to explain why it is that life took this shape that it actually has done. I just need to say thank you to a lot of wonderful students who've been working in the lab and thinking about these questions and helping me think about these questions over the last few years, and thank you very much.